We're recording with the Jones's AP Bio Spectrophotometry Lab using chloroplast solutions. This is a chloroplast solution. We just made it. It's just blended spinach in 0.13 molar sucrose solution. And we're under a grow light right now. And these are our 4 experimental cuvettes. Well, really, the first one is the control, a negative control. It's called the blank. And these 3 are the experimental ones. The first one, I have 4 mLs of water, 1 ml of phosphate buffer. And the rest of them, I have 3 mLs of water, 1 ml of phosphate buffer. So the first one has 1 ml of volume extra. Why? Because we're not putting DPIP in that first one, okay? We're not putting DPIP in the first one. The first one does not get DPIP. You'll see why in a second. Let's do the first one. Let's do the blank. So chloroplast suspension. This is the blank right here. I'm going to take some chloroplast suspension. I'm going to add 1, 2, 3. Now I know those are drops. I realize there's some error there. And we could do microliters using a pipette, but it's okay. It still pretty much works. The drops are pretty accurate because this is still a blend anyway. It's not like it's perfectly homogeneous. So one of the most important things is we invert these cuvettes. Notice how I'm holding them from the top, put my finger over it, and I'm going to invert it. I'm never touching this part. You can see it's not homogeneous, and I'm going to invert twice, always twice. If you see me forget to do that, yell, okay? I have to invert it, because if I don't, then things will naturally settle. Big components will settle, because nothing here is really dissolved. It's kind of suspended. And the light goes through roughly 25% of the way up the cuvette. So if things are settled, the light will go right above it. Okay, so here we go, invert twice. 1, 2, that ensures that they're mixed. Okay, now I'm gonna put it in the spec. This is the blank again. Put it in the spec. Almost made a mistake. I'm zeroing it out. Okay, it is zeroed. The spec is zeroed. I put it in, and I put it in so you guys can see, and I'm setting 100% T. Setting 100% T. So 100% transmittance. What I'm saying is that even though it doesn't, 100% of the light goes through this cuvette, the blank. It means that I'm canceling out the water and the phosphate buffer and the chloroplast. I'm canceling those out. I am quote unquote blanking them out, canceling them, because I don't care about them. I care about the DPIP. That's what I quote unquote care about. That's what I'm measuring. So Sim, the great Sim, Snow Sim, is my timer. Sim, this is time 0. That's time 0. All right, here we go. We're gonna work fast. This is the darkness one. I'm gonna get the DPIP. I'm gonna add 1 ml of DPIP. You can see it's blue to my darkness cuvette. And I'm going to add 3 drops of chloroplasts. 1, 2, 3. I'm going to take the darkness cuvette out. You can see how blue it is. See, it's green and clear in the bottom, blue in the top. It's not mixed. We know that. So I'm going to invert it twice. Here we go. Okay, Danny is my timer. Danny, I have 17 transmittance. 17 transmittance for the darkness. I have to work fast because we've started the time for time 0 already. DPIP to number 3. 1 ml of DPIP to number 3. That's my living light. This is living light. See how it's not mixed? 1, 2. 15% T. 15% T, Danny. 50% T for living light. And lastly, boiled. One ml of DPIP. These are boiled chloroplasts. They're dead. 1, 2, 3. Two inversions. %16 T, Danny. 16% T. Okay, that was time 0. How we doing on time, Sim?
Good. We're at 2 minutes and 23 seconds.
Okay, question for you guys. Did I forget to add chloroplast to my living light?
Yes.
I did. Thank you. I knew it. That's okay. Adaptation. Here we go. Living light, Danny. 1, 2, 3. Whoever said yes, good job. I thought I did. I thought I forgot. Here we go. Living light, Danny. Time 0. Redo. That's okay. 12%. 12%. So we'll do that one last each time. No big deal. Okay, now we wait. If you're watching this video, you might want to fast forward a tiny bit. You might want to fast forward about 2 minutes. I'm going to move living light 1 back to remind me to do this one last. That's living light. My darkness will be back here. So I'm going to go blank, darkness, boiled, living light. Doesn't matter the order, as long as you do them at the time. And as long as they have roughly 5 minutes between. So the chloroplasts now should be photosynthesizing to some degree, light reactions, sending electrons to NADP+. But this blue stuff, DPIP, is grabbing the electrons and should be turning clear over time. And that's what we're measuring. The disappearance of DPIP. How's our time, Sim?
We are at 4 minutes and 15 seconds.
All right, got 45 seconds. So that's what we're measuring, guys. We're measuring the disappearance of DPIP over time. The turning of the DPIP from blue to clear because it becomes reduced with electrons from the photolysis of water. The photolysis of water sends electrons throughout to ETC at the thylakoid membrane to NADP+, but the DPIP grabs those electrons. Okay, so I'm getting ready for my 5 minute. I re-zero the spec.
All right, we're at 5 minutes.
Okay, 5 minutes. Here we go. Re-zero the spec. Here's the blank. Inversion. I'm resetting 100% T. 100% transmittance. So I'm telling everything to go through this cuvette. I'm saying essentially 100% of the light goes through this cuvette. It doesn't. You know, the water, the phosphate buffer, the chloroplast, those all grab and absorb light. But I'm canceling them out, blanking them out. So we can just measure the variable in the other three, and that's the DPIP. Here we go, the cowboy. Cowboy comes out, invert twice. Everything gets the same treatment. We put this in, and we have 23% T, 23% T for darkness. Thank you. You're welcome. All right, 23% T for darkness. Now I know I'm going to skip over. I'm going to the boiled now because I realized that I did this one later. Going to the boiled. Here we go. Boiled. Boiled, Danny, is 15% T. 15% T for boiled. Okay, here we go. Living light. Living light, Danny. 2 inversions. (27% T for living light, Danny. 27% T for living light at 5 minutes. Okay. So now I assume we have somewhere around 3.5, 4 minutes remaining. So to reiterate, this is the blank. That one's the blank. It has water, chloroplast, phosphate buffer. But I blank those out. I cancel them out because I don't necessarily care about them, meaning I don't want to measure what happens to them. So I'm canceling those out. That's why I set 100% transmittance for the blank. The remaining 3, the darkness, the boiled, the living light, those all have DPIP. That's the variable that's changing. The DPIP becomes reduced with electrons. So that should go clear. And that's why the percent transmittance should increase over time. It should increase over time. And we'll see. Now, will it increase in the darkness, right? Well, it's in the darkness. Probably not. That's right, cowboy, probably not. Will it increase in the boiled? Well, he can't speak because he's boiled. Totally dead. Will it increase in the living light? Well, it's living in the light. How's our time, Tim?
We're at about 8.5 minutes.
Okay, 8.5. Here we go. So this said, in terms of the period, guys, we're gonna finish this. Then we'll take a break. All right, promise. I'll take a break. I just can't take a break at the bell, the midway. All right, we'll take a break as soon as we're done. I promise. All right. And if I forget for tomorrow's asynchronous day, you do not have to come to class. Pretty sweet, right? You do not have to come. I will take attendance and I'll mark you present because you'll be here because you'll be working on the assignment. And I'll be here the whole time. If you have any questions, you can pop in. I'll keep my cam on. I'll keep it going. You just yell out. But that's optional.
That's 10 minutes, Mr. Jones.
That's 10 minutes? Okay, here we go. Here's my blank. 2 inversions. Almost perfect. Nothing's really changed in that. It's still 100% T. All right, so I just set 100% T for the blank. Let's do the darkness. Come on. Here we go. Darkness. Okay, here we go. Darkness, 22.
Got
20.
it. Check out The Darkness Station on YouTube.
Oh. That's my Google Home. Nice job, Google Home. Bad job. Bad Google Home. 22%. Darkness. Okay, Danny? Here we go. Boiled, Danny. Boiled. 2 inversions. 2 inversions. We're looking at 17% for boiled. 17% for boiled. And lastly, living in the light. the living in the light, 2 inversions, 38%, 38% T. Living in the light, 38% T. Whoops. Okay, good. So if you guys have been watching, which cuvette is increasing in transmittance? They're all being coy. They're all being shy. Yes, if you've guessed, it's this one, the living in the light, which makes a lot of sense because nothing's boiled in here. Nothing's in darkness. There is chloroplasts. There are chloroplasts and there is DPIP. So over time, those living chloroplasts should be making, splitting water, sending the water to NADP+, but the DPIP is grabbing those electrons and is becoming reduced and is turning clear. That's the whole point. So I've been casually watching this. I'm not even looking at the data. I know Danny is. She's recording for us. And this one, the living light one, seems to be working and doing what it should be doing. If you're wondering sources of error, when I take the close off the cowboy one, the one in the darkness, yes, you're right. It's exposed to light for like probably 5 seconds. And you're right, that's the source of error. I probably should turn the light off temporarily, but then I'm affecting the others. The darkness one, maybe I should keep that off to the side, but it's not super practical to do that. So, but you're right, that's the source of error. How's time, Sim?
Good, we're at 13 minutes and 35 seconds.
Okay, it's 1.5 minutes. So one last thing before we do the 15-minute, because we're almost there. This is something I didn't mention yet. I'm using 605 nanometers. The dial I haven't shown you yet, 605 nanometers. I'm using 605 because the DPIP absorbs 605. Now it absorbs other wavelengths well, but the DPIP absorbs 605 nanometers really well. So if there's a lot of DPIP, and I mean oxidized DPIP, blue DPIP, if there's a lot of blue oxidized DPIP, then it will absorb more and transmit less. If there's less oxidized DPIP, therefore, if it's more clear, it'll absorb less and transmit more, allow to transmit more. So that's why I'm using 605, because DPIP absorbs 605 nanometers. It absorbs others, but it absorbs 605 pretty well. And 605 is somewhere towards it's more towards the red, as you know. It's pushing towards red. And that's why this is blue. You know, blue is more this is blue. Blue is more towards the 400s, 500s. Okay, our final one's coming up. We're going to stop the video as soon as we're done with this one.
That's 15 minutes.
15 minutes, very good. Here we go. Final one. 2 inversions. This is the blank. Set 100 T. I've set 100 transmittance. Nice. This is the darkness. He doesn't like getting undressed. It's in the name of science. Here we go, darkness. The darkness is 20% T, 20.0% T for darkness. Danny, 20% T for darkness. Boiled, Danny, here we go, boiled. 2 inversions. For boiled, 17.5% for boiled at 15 minutes. And lastly, our living light, I'm stalling a bit because I know this one was stalled a bit. Our living light, charged up in the light, living light. Again, I'm stalling because I had to stall a bit for this one because I put this one in last. They probably didn't get as much time. 2 inversions, living light. Here we go. Final reading, living light, 15 minutes. We are at 48.5%, 48.5% for living light. And it's over. Notice that I only added chloroplasts at time 0. I didn't keep adding chloroplasts. I added only at time 0. And the one variable throughout all these is the DPIP, DPIP, DPIP. The blank had no DPIP. We blanked out everything in there except for the DPIP. All right, and that's the video. You guys can see your respective data tables in your respective classes. I'm going to stop recording. Stop recording. Yes.